Assalamualaikum dan salam sejahtera. This is class for General Biology. I'm Dr. Shaida and I will be your instructor for this course. We are in chapter 3 in the topic of chemical component of cells. Next, let's look on the fatty acid. Fatty acid is an important component of lipid which is present in the living cells, and it becomes main component of plants, animals, and microorganisms. Generally, fatty acid will consist of straight chain of hydrocarbon that attach to one carboxyl group at the other end. The hydrocarbon is hydrophobic and not very reactive chemically. Nevertheless, the other is carboxylic acid become very hydrophilic and chemically reactive, and it will form carboxylic acid as it is in aqueous solution. And this makes the fatty acid to have both hydrophobic and hydrophilic component. The hydrophobic hydrocarbon is attached to the hydrophilic carboxylic acid group, and different fatty acid will have different hydrocarbon tails. Hundreds of different kinds of fatty acid exist. Some have one or more double bond in their hydrocarbon tail and are said to be unsaturated. Fatty acid with no double bond are saturated. The hydrocarbon tail of the stearic acid is saturated. It has no double bond between its carbon atoms and it contains the maximum possible number of hydrogen. Some other fatty acids, such as stearic acid, will have the unsaturated tails with one or more double bond along their length. The double bond creates kinks in the hydrocarbon tails, interfering with the ability to pack together. The presence of double bond accounts for the difference between hard and soft lipid. Fatty acid tails, however, are also found in cell membrane where their tightness of the packing affects the fluidity of the membrane. Many different fatty acids are found in cell differs only in their length of their hydrocarbon chain and in the number and position of the carbon-carbon double bond. Fatty acids serve as a concentrated food reserve in the cell, as they can be broken down to produce about 6 times as much usable energy weight for weight as glucose. Fatty acids are stored in the cytoplasm and many cells in the form of fat droplets composed of triacylglycerol molecule, which is the compound that is made of 3 fatty acid chains that covalently join to a glycerol molecule. The triacylglycerol are present in the animal fats that is found in meat, butter, and cream, and the plant oil such as the corn oil and olive oil. When a cell needs energy, the fatty acid chains can be released from the triacylglycerol and broken down into carbon units. These 2 carbon units are identical to those derived from the breaking down of glucose, and as they enter the same energy yielding reaction in order to produce the energy. The most unique function of the fatty acid is in the formation of the lipid bilayer, which is the basis for the all cell membrane. These thin sheaths, which enclose all cells and surround their internal organelle, are composed largely made from the phospholipid. Like the triacylglycerol, most phospholipids are mainly constructed from the fatty acid and glycerol. In this phospholipid, however, the glycerol is joined into 2 fatty acid chains rather than to 3 as in triacylglycerol. The remaining hydroxy group on the glycerol is linked to the hydrophobic phosphate group, which in turn is attached to a small hydrophilic compound such as choline. With their 2 hydrophobic fatty acid tails and hydrophilic phosphate containing head, phospholipids are strongly amphipathic. These characteristics of amphipathic composition and shape give them different physical and chemical properties from triacylglycerol, which are predominantly hydrophobic. In addition to the phospholipid, cell membrane contain different amounts of other lipids, including glycolipids, which contain 1 or more sugars instead of a phosphate groups. The amphipathic nature makes the phospholipid readily form membranes in water. This lipid will spread over to the surface of water to form a monolayer with their hydrophobic tail facing their air and their hydrophilic heads in the contact to the water. Such the two molecule layer can readily combine tail to tail in water to form the phospholipid sandwich that is the lipid bilayer. Formation of long hydrocarbon chain with 4 linked aromatic rings will form steroid, which is cluster of compound under lipids. Lipids are closely defined as molecules that are insoluble in water but soluble in fats and organic solvents such as benzene. Steroids are also involved in the cell communication. In cells, steroids serve as the precursor for hormones, including estrogen, testosterone, and cholesterol, which regulate the human body system. Then we go further on the amino acid. Amino acid is small organic molecule with one defining property, which is they are all possess one carboxylic acid group and one amino group that are both linked to the alpha carbon atom. Each amino acid will have a side chain attached to its alpha carbon. The identity of this side chain is what that distinguishes one amino acid from another. There are 20 kinds of amino acid present which serve as the protein monomer. Each amino acid consists of center carbon atom that is bonded to 4 covalent partners. The component of amino acid will consist of carboxylic group, amino group, and hydrogen atom. Normally, radical group is attached to the 4th bond of the center carbon. There is a unique side group that is giving attached to the amino acid that giving special chemical properties for each amino acid. Amino acid can be classified into nonpolar, polar acidic, polar basic, and polar neutral. The following rules, with 2 exceptions, can help you to identify the class of amino acid according to its type. For the non-polar amino acid, they contain the aliphatic hydrocarbon chains or aromatic rings. And the polar acidic amino acid, it will contain one carboxylic acid or carboxylate group in the side chain or R group. This is in addition to the one in the backbone of the amino acid. Polar basic amino acid will contain one amine that can be neutral or charged in their side chain. This in addition to the other one in the backbone of the amino acid. For the polar neutral amino acid, it will contain one hydroxyl group, one sulfur group, or amide group in the R group. Amino acid has Zwitter ion characteristic, which is having salt-like behavior. In physical characteristic, amino acid is crystalline solid with relatively high melting point, and most are quite soluble in water and insoluble in non-polar solvent. In solution, the amino acid molecule appear to have charge with changes with pH, and intramolecular neutralization reaction lead to a salt-like ion, which is called as Zwitter ion. Cell use amino acid to build protein, which is the polymer which made from the amino acid that are joined from head to tail in long chain that folds up into 3-dimensional structure that is unique to each type of protein. The covalent bond between the 2 adjacent amino acid in protein chain is called as peptide bond, and the chain of amino acid is also known as polypeptide. Peptide bonds are formed by condensation reaction that link one amino acid to another. A protein can be built from a set of only 20 amino acids, each of which has a unique side chain. The side chain of amino acid will have different chemistries. The largest group of amino acids have non-polar side chain, and several other amino acids have side chain with positive or negative charges, while others have polar but uncharged side chain. Due to the complexity of protein molecule that has been made up from 20 different kinds of monomer, it can be classified by its structure. Long protein backbones can adopt unique 3-dimensional secondary and tertiary structure, which both of which are dependent on the primary structure. When two or more protein strands of protein are held together by the intermolecular process, they can also adopt specific quaternary structure. Let's look into different types of protein structure. The first structure is the primary structure. In this structure, the protein sequence is written from the end with a free amine group, or known as N-terminus, to the end with a free carboxyl group, or known as C-terminus. Next is the secondary structure. A protein secondary structure is the three-dimensional shape of small segments of protein. In secondary structure, amino acids that are not next to each other along the backbone are close enough to each other for the hydrogen bonding. The type of secondary structure is defined by the pattern of hydrogen bond between amide groups that are not adjacent in the protein backbone. Two common elements of secondary structure which are present in over 60% of known protein are the alpha helix and beta sheath. The tertiary structure of protein is the overall three-dimensional shape of the protein. It is how the alpha helix, beta sheath that modify the protein together in order to form the overall structure. The tertiary structure are often also defined by various intermolecular interactions involving the protein side chain as well as the environment in the protein. Two general kinds of protein are found in cell, whether it could be water-soluble protein and water-insoluble protein. Water-soluble protein include the enzyme and transport protein that are mostly found in the pre-cellular compartment such as the cytoplasm, nucleus or endoplasmic reticulum. In the tertiary structure of most water-soluble protein, most dihydrophobic side chain groups are in the interior of the protein and away from the water, while most polar side chain groups are mostly capped on the exterior and therefore are exposed to the water. The backbone amide group, while polar and capable of forming hydrogen bond, are not interacting much with water because they are always engaged with the hydrogen bonding from one to another in the alpha helix and beta sheath. Besides, it also involves different types of bond that include the covalent bond, hydrogen bond, ionic interaction, hydrophilic and hydrophobic interaction. Quaternary structure arises when more than one protein chain is present and the chain form larger structure. For example, the hemoglobin in blood, which is composed of 4 chains as observed in the picture, colored magenta, blue, aqua and tan. When oxygen binds to one subunit of hemoglobin, the interaction between the subunit change. In other words, oxygen binding affects the quaternary structure. The quaternary structure is the usual mechanism for the process known as allostery, in which the binding of molecule at one site on the protein has effects at the other site that away from that site, and cooperatively, the binding of molecule at one site in multi-subunit protein increase or decrease that the likelihood that another molecule would bind at another site of the protein. The cytoplasm of cell is highly structured due to the aid of protein, particularly in the eukaryotic cell, which tend to be bigger and need more mechanical support than the prokaryotic cell. An extensive network of filament that consists of microtubule, actin filament, and intermediate filament can be detected with variety of microscopic methods. Microtubule, for example, play a major role in organizing the cytoplasm and in the distribution of organelles. They also form during the mitotic spindle during cell division. Actin filaments are involved in various forms of cell movement, including cell locomotion, contraction of muscle, and cell division. In the intermediate filament are strong fibers that serve as architectural support inside a cell. Cells also rely on thousands of different enzymes to catalyze metabolic reactions. Enzymes are proteins and they make biochemical reactions more likely to proceed by lowering the activation energy of the reaction, thereby making this reaction proceed thousands or even million times faster than they would do without a catalyst. Enzymes are highly specific to the substrate. They bind to the substrate at complementary areas on the surface, providing snug fit that many scientists compare to as lock and key model. Enzymes work by binding one or more substrates, bringing them together so that reaction can take place and releasing them once the reaction is complete. In particular, when substrate binding occurs, enzymes undergo conformational shift that orient or strain the substrate so that they are more reactive. The protein in the plasma membrane typically help the cell to interact with the environment. For example, plasma membrane protein carry out function as diverse as varying nutrients across the plasma membrane, receiving chemical signals from outside the cell and translating the chemical signal into the intercellular reaction, and sometimes also involved to anchor the cell in particular location. The fourth component in living organism is nucleic acid. DNA and RNA are built from subunit called as nucleotide. Nucleosides are made of nitrogen-containing ring compound linked to a 5-carbon sugar which can be either ribose or deoxyribose. Nucleotides are nucleosides that contain one or more phosphate group attached to the sugar, and they come in two main forms. Those containing ribose are known as ribonucleotide, and those that containing the oxyribose are known as deoxyribonucleotide. The nitrogen-containing rings of all these molecules are generally referred to as bases for historical reasons, since under the acidic condition they can each bind to the hydrogen ion and thereby increase the concentration of hydroxyl ion in aqueous solution. There are two families of nitrogenous base, either pyrimidine or purines. Under the pyrimidines group, cytosine, thymine, and uracil have single 6-membered ring, whereas for the purine, adenine and guanine have 6-membered ring that fuse to a 5-membered ring. We need to remember that the sugar that is forming the DNA structure is made from deoxyribose, whereas for the RNA, the sugar is made by ribose. The nitrogenous base in DNA pair that made up through hydrogen bond, it will make the complementary base pairing, in which the adenine will always be paired with thymine and the guanine will always be paired with cytosine. The complementary pairing can also occur between two RNA molecules or between parts of the same molecule. In RNA, however, the thymine will be replaced by uracil, so the uracil will be paired with the adenine.
DNA, the sugar is deoxyribose, whereas in RNA, the sugar is ribose. The nitrogenous bases in DNA pair up and form the hydrogen bond. Adenine will always pair with thymine and guanine will always pair with cytosine, and this pairing is known as the complementary base pairing. And the complementary pairing can also occur between two RNA molecules or between parts of the same molecule. However, in RNA, the bases of thymine will be replaced by uracil, so the adenine will pair with uracil. The double helix DNA is related to genomics, which is the term used to describe the physical structure of DNA. A DNA molecule is made up of two linked strands that wind around each other to resemble a twisted ladder in a helical shape. Each strand has a big bond made of alternating sugar, or known as deoxyribose and phosphate groups. RNA is ribonucleic acid that helps in the synthesis of proteins in our body. This nucleic acid is responsible for the production of new cells in humans. It is usually obtained from DNA molecule. RNA resembles the same as that of DNA, but the only difference is it has single strands, which is unlikely to DNA, which is having two strands. RNA consists of only one single ribose sugar molecule in it, thus making the name as ribonucleic acid. RNA also referred to as an enzyme as it helps in the process of chemical reaction in body. Nucleic acid is known to be found in all life forms as well as some non-living entities such as bacteria, archaea, and even found in the organelle such as mitochondria, chloroplast, and also in the virus and virus, which is the plant pathogen. All living cells contain both DNA and RNA, exception to some cells such as mature red blood cell. And even the viruses also contain either DNA or RNA, but usually they not contain both type of nucleic acid. Nucleic acid function as heredity in cell. When a cell divides, its DNA is copied and passed from one cell generation to the next generation. DNA contains the programmatic instruction for cellular activities. When organisms produce the offspring, this instruction is in the form of DNA are passed down. RNA is involved in the synthesis of protein, which is the typically information will be passed from DNA to RNA, and from RNA will be synthesized into resulting protein. In summary, the cell composition of living organism primarily consists of four essential elements, which are the carbon, hydrogen, nitrogen, and oxygen. These elements are the building blocks of biological molecule and are crucial for the structure and function of cells. Carbon is the fundamental element of organic molecules. It forms the backbone of biomolecules, including the carbohydrates, lipid, proteins, and nucleic acid. Carbon atoms can form strong covalent bonds with other carbon atoms and various elements, allowing for the diversity of organic compounds in living cells.
That's all for today's class. Thank you for listening. See you in the next lesson. Bye bye. Assalamualaikum warahmatullahi wabarakatuh.